Did you know? Identical twins are born randomly all over the world, but fraternal twins are more likely to be born in some populations than others. They are most common among people of African descent and least common among people of Asian descent. And mothers over 40 are also more likely to bear fraternal twins.
Each molecule of DNA contains the unique recipe for making a living thing. Today, scientists can use this recipe for many purposes, from cloning animals to solving crimes.
What do you know about DNA?
When I think of DNA, I think of the genetic makeup of a person. DNA defines who a person is because everybody has a unique DNA.
Your parents' DNA is passed down to you through chromosomes.
It's like the code for your body. It tells you what you're gonna look like. DNA, it's a genetic makeup of a person arranged in a helix form, double helix.
It helps identify who a person is.
I know scientists can use it to match up blood samples and match up people using DNA.
They use DNA for cloning.
DNA are like genes, and right now we're using them to genetically modify our foods.
DNA is deoxyribonucleic acid.
For the first time in history, it is within our power to intervene in the design of life. Breakthroughs in genetics are bringing us closer to what we used to call science fiction. The age of genetics is upon us. The ability to clone animals, grow organs, and cheat death is within our grasp. Will we duplicate ourselves, alter our genes, and dare to design immortality? Science will continue to revolutionize the world.
In the next 100 years, we're going to have an incredible understanding of genetics. Inevitably, we're going to have sequence information from many different species and many different individuals. It is information that, to me, is probably one of the most important breakthroughs in human history.
With the advent of powerful computers, scientists explore the inner space of our genetic code.
We've now developed telescopes that let us look at DNA at an incredible level of detail. And within a few years, we're going to know the sequence of 3 billion bases of DNA. Once you have that information, there's a lot more interest in genes because now we know what genes are. We know what all of the genes are.
It's information that redefines our concept of mortality and may extend life indefinitely.
I don't think that people are born to die. We have in ourselves the fundamental essence of what a human being is, or any animal or plant or living thing is an immortal molecule, DNA.
In the beginning, the first life known as the original ancestor was a one-celled organism that contained genes in short strings of DNA. It had the miraculous ability to make rough copies of itself. The DNA molecule of that original ancestor has passed through 3,000 million organisms to become dinosaurs, trees, chimps, and people. The living world has infinite variety because every time the DNA copies itself, it changes slightly. This is evolution.
DNA is a molecule that has two parts to it. When it divides, the two strands separate, and two new strands are created. You can't tell what was old and what was new. So it is, in fact, the same molecule. That process has gone on for 4 billion years. The DNA that's in you, the DNA that's in me, has a direct trace right back to that molecule that was on Earth 4 billion years ago. And you can't say that either this molecule or this one was the original.
Every DNA molecule forms a twisted ladder with rungs of 4 specifically paired chemical bases. Adenine pairs with thymine and guanine with cytosine. The chemicals dictate the creation of different proteins, which influence everything from eye color to IQ. Recent breakthroughs in the mechanics of genetics have revolutionized how criminal investigations are conducted, even when a case is decades old. In 1954, Marilyn Shepard was murdered in cold blood. The crime captured worldwide attention and spawned the TV series The Fugitive. If modern genetic technology had been available, Sam Shepard would have met a different fate.
All of Cleveland awaits an answer to the question, who murdered Marilyn Shepard?
It was July 4th, 1954. Sam Shepard had guests over. He had, I guess, a few glasses of wine. He went to sleep under the stairs. His wife went to bed. A visitor came by, thinking Sam was away because he was not in plain view. And Sam heard some noise, got up, grappled with somebody. He called him a bushy-haired man.
The jury did not believe Sam Shepard's story. He was convicted and served 10 years in a state penitentiary for the murder of his wife. In 1966, Shepard appealed his conviction with the help of the trial lawyer F. Lee Bailey.
I'm confident that Dr. Sam will never be retried because the state does not now have a case it never did have.
In fact, Sam Shepard was retried, but Bailey was able to get him acquitted on a technicality. Still, many people believed he was guilty. He lived under a cloud of suspicion until his death in 1970. His son was determined to prove his innocence, but he made no progress until science began solving crimes.
DNA is a silent witness. It doesn't talk, but the scientist's job is to find out the facts, what the evidence tells.
Dr. Muhammad Tahir, a pioneer in DNA technology, was asked to help solve Marilyn Shepard's murder. To make the case, Tahir needed to compare DNA from the blood-stained items at the crime scene to a sample of Sam Shepard's DNA. But Shepard was dead and buried. Where could he get the DNA? Tahir came up with an ingenious way to solve the problem. He scraped the back of old postage stamps on love letters that Shepard had mailed to his wife in the mid-1940s. The tedious process took hundreds of hours. Once a DNA sample is obtained, scientists analyze its X-ray. The black bands show the patterns of the chemicals that make up the rungs of the DNA ladder. This fragment of DNA was found at the crime scene. And this DNA sample comes from a suspect. If the two match, there is virtually no chance for error, as long as the sample is not damaged or contaminated. In the Shepard case, the DNA sample was insufficient, but there was enough evidence to warrant taking another step. The authorities exhumed Shepard's body, and Tahir gathered more DNA. When he compared it to the DNA from the evidence, Tahir confirmed that genetic material at the crime scene did not match either Sam or Marilyn. That could only mean one thing.
In this case, the DNA found on the wood chip and on the Sam's pants did not belong to Marilyn Shepard, and neither it does belong to Sam Shepard. It has to come from somebody else.
Sam Shepard had been telling the truth when he claimed another person in the house was the murderer. The real killer was never tried, but Shepard has been exonerated, his family name cleared.
It's regrettable that it took 45 years for Dr. Sam Shepard to be vindicated. DNA evidence properly handled, tests properly conducted, is extremely reliable. The odds are astronomical that it was or was not this person. So it is a definitive way of excluding people, and that has been its principal benefit.
DNA evidence began to play a vital role in criminal investigations, beginning in the early 1980s. Today, standard police procedure for every violent crime includes scraping crime scene items for DNA. The FBI considers DNA evidence its most powerful tool, and now it is using it in a new way. In 1994, the FBI developed a software program they dubbed CODIS, which greatly increases the number of DNA matches nationwide.
In the past, typically, laboratories match a known sample against something they found at a crime scene. Looking at various points on that evidence, they say, yes, the known sample matches the unknown, or it doesn't match. That can be on a bullet, a cartridge casing, a fingerprint. The process is the same. What CODIS does is it adds a little bit extra. You don't need to have that known sample to solve a crime with CODIS. You go in there and search DNA evidence found at a crime scene, and without having any known samples, you can search the database and get a list of possible contributors of that sample.
In a matter of minutes, CODIS can cross-reference hundreds of DNA samples of known criminals to DNA found at a crime scene. When it makes a match, it's called a cold hit. The criminals have been fingered by their own genes. CODIS has led to the conviction of more than 300 criminals, including these.
We think ultimately CODIS is going to be just like fingerprints. Obviously DNA, there's much more you can do with it than just a fingerprint, but for law enforcement purposes, it's used just like conventional fingerprints.
As the technology has gotten cheaper, it has become more widespread. Someday, the FBI may only need to click a mouse a few times to catch a thief. This is Dolly. When it comes time to write the history of our age, the creation of this little lamb will stand out. Dolly was the world's first clone created from an adult cell of a living animal. Before Dolly, geneticists believed that once a cell becomes specialized, like a skin cell or a brain cell, the genetic instructions for creating another life were permanently switched off. The creators of Dolly forced cells into hibernation by starving them of some nutrients. This starvation essentially took the cell's genetic material backwards in time to a state that could create life. It was a moment that altered our notion of life and how it can be manipulated.
It really turned things on its head because all the data suggested that this should not work. And all of a sudden you see that it happened and it has worked, and it really makes you rethink the entire system and what's going on.
So how do you make a clone? It starts by gathering DNA from fetal cells, embryonic cells, or even the skin of adult animals. Specific cloning techniques are changing fast, but the results are the same. One of the most successful cloning methods happens in a room kept at about 44 degrees Celsius, the same temperature as a cow's uterus. The actual creation of the clone happens in a petri dish underneath the powerful lens of a microscope. With the guiding arms of the micromanipulator, the nucleus of an egg cell is sucked out, which means the egg has no genetic instructions. Then the DNA from another cell is inserted into the egg. A jolt of electricity jumpstarts the genetic material into creating life. But to make it into the world, the clones need the help of a surrogate mother.
In this device right here is a transgenic embryo that we made in the laboratory last week. And also designed to go along with this unit is a plastic sheath. Now this device will be used to deliver the embryo up into the uterus of the cow.
After the transfer, the growing clones are carefully monitored. Only 1 in every 95 pregnancies results in the live birth of a clone. Until then, geneticists take on the role of nervous mothers, carefully checking progress with routine sonograms.
Looks real good. That's a real solid, healthy 2.5-month pregnancy. Looks very good. Probably one of the neatest things to observe is the heart beating.
This fetus is a genetically altered cow clone. If it is taken to full term, it will be an offspring of genetic engineering, not bovine love. Geneticists have gone beyond sexual reproduction. They've invented a new way of conceiving life.
To tell you the truth, I've never once ever thought that I was a creator. I feel more like a manipulator of taking a very complex, a very beautiful system and manipulating it in ways that it can be of benefit to mankind.
Scientists may not feel all-powerful in their work, but technically, they are creating some new life forms.